This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 388, recorded on May 6th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,400 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
Here in New York City.
Right here in cloudy, cold, rainy New York.
It's terrible. 10 degrees Celsius.
It reminds me of the summer I spent at the Trudeau Institute up near Saranac Lake, New York. I was doing some research up there one summer. Just so happened that was the summer of the Summer Olympics in Montreal. It was not a summer. It was like this out there every single day. It rained for 40 days during the summer.
My weather app shows an active weather alert, and it's in all caps. They haven't yet switched to the new system.
Probably pay attention to that one.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's pretty similar up here and has been all week and is supposed to be pretty much like this. We were supposed to get a brief respite on Monday and then back to this.
Oh.
Winds out of the east, which is never a good sign.
I show it getting nice on Sunday, Monday, Tuesday, Wednesday, Thursday. Here, can't be that different though.
Oh yes, it can.
They're saying back to Monday's supposed to be nice, and then Tuesday partly sunny, and then back to chance of precipitation 30-40% through Tuesday, Wednesday, Thursday.
Alan, you get a lot of weather deflected across the Adirondacks. We don't get that.
Yes, we get two separate streams of weather systems. We get
Green
the stuff
Mountains.
that comes in from the lakes to the west, and we also get the stuff that comes up the coast, and they sort of battle each other right around this area.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. The weather's great here. And it was great yesterday. And it's going to be great on Sunday. It might rain tomorrow, though. But right now, it's 68 degrees Fahrenheit, 20 degrees Celsius, sunny, just fabulous.
Today, this morning, I had a group of middle and high schoolers visit my lab.
Cool.
Like 12 kids, right, Dickson?
It was fun.
I'm exhausted.
So are they.
It was my son's private school.
No, you should have heard the questions from these kids. It was just amazing.
Some of them had such good questions.
They were
One
amazing.
kid in 5th grade.
Yeah, this guy was.
Little guy.
M&M, I
oh
call
my
him
gosh,
M&M.
he was so smart.
Yep.
It was just, I can't even remember any of the questions, but they were great. They were all really, they don't get to see a lab or
Precocious was
anything like this. So they were all excited. And Dickson, they all crowded around Dixon's desk, and he was showing them worms and posters. And it was just great. We had a good time. Yeah, but I am really tired. I don't know. Dealing with kids is exhausting.
How would you like to do that every day, teaching?
So I was telling them my story. I said, I never wanted to be a doctor because I don't really want to take care of people. I just take care of my own kids. And Devin said, no, you don't. Oh, boy.
Danger.
I shouldn't have said that. Well, we would like to congratulate the newest inductees into the National Academy of Sciences.
Uh-huh.
Sorry, you and I didn't make it, Dickson.
Not again?
We will say that every year until we die.
We will.
Joe DeRisi, who was on TWiV 196, Eugene Koonin, TWiV 275, and XJ Meng, TWiV 285.
Outstanding.
And over on Twitter, SciGuy999 posted this with the episodes, and he said, the TWiV bump.
Nice.
We congratulate everybody who got that, but especially the ones who got in with
another virologist who got inducted is Skip Virgin. We talked about his work on TWiV 204. Although I did ask Skip years ago to come on TWiV, and he said no. Yeah, he said no. But he got the bump anyway, you see.
That's right.
Even if you're asked.
It was the placebo effect.
It's an honor just to be nominated.
That's right.
Was that the placenta effect?
Placebo. I said placebo.
So why didn't he want to come on?
Yeah.
He
He
told
thinks we're
me,
muckraking or
no,
something.
no. He said, I don't want to have to worry about what to say and what not to say. Isn't that ridiculous? I think
We
that's
He
don't.
absurd.
yeah,
Why
he
should
might
he?
change his
We don't
mind
worry.
by now. Yeah.
I think you should just talk about science. What do you care?
Yeah.
Don't worry about being scooped.
Hey, maybe he'll change his mind now that he's in the National
I've been
Academy.
scooped so many times. Look at me. I'm fine. Or maybe,
Well, and if
maybe
Skip
not.
does come on now, I mean, where do you go from National Academy? He's looking at a Nobel Prize with the next TWiV bump.
There you go.
Yeah, so congrats to all those. And we have some follow-up. We actually have a lot of email we will have to catch up with, but the first one, Alan, can you read Ted's, please?
Sure. Ted writes, Hi, Vincent. I wanted to thank you again for featuring my paper on TWiV 386 and having me on to discuss the research. I had a great time doing so. Since the taping, I realized that during our discussion, I forgot to mention one thing that I wanted to pass along to your listeners. That is, how much of a pleasure it was to publish through eLife, and not just because they decided to publish my work. Kartik Chandran mentioned this during his TWiV 05 episode, and I wanted to join him in praising the publication process through this journal. Several aspects of the process were more streamlined than those of other journals. That said, the initial submission process is so much easier and more streamlined than other journals that I felt they should be praised for it specifically. I can't overstate how nice it was to upload a simple compiled PDF file for review as opposed to spending an entire afternoon or longer figuring out which files or figures are being rejected by the online submission compiler and then determining the reason behind the failure. I understand that each journal has their own formatting requirements, but why is it necessary to worry about that prior to review? In my opinion, it's an entirely pointless waste of time to have to deal with this until the manuscript has been accepted and those issues become relevant, especially if the manuscript is rejected. The publishing process would be much less of a headache if every journal had a similar system. Okay.
I do agree that when you have to...
That's Ted Deal, who's a postdoc at UMass.
Yeah, who was our guest a couple weeks ago. I do agree that, a lot of these journals, I know the ASM journals, you have to submit every figure separately, and it checks it, and it takes a long time. This is great. Just send a PDF. I think that's wonderful.
And it's a perfectly valid point that in the initial process, I mean, it needs to be triaged and then reviewed.
Yeah.
And why do you need a file that's perfectly usable for publication if you're going to reject most of them?
Sure. I had actually meant to ask him how he liked the eLife process, and I didn't, but I'm glad he wrote in because we're doing more and more papers from eLife, and sounds like it's good. Sounds like they really want to publish science.
Yeah.
As opposed to making a profit, maybe, and so forth. The next letter is from Mehul Suthar, who wrote, this came in last week, and I didn't read it because he had written it actually in response to a blog post that I had put up at Virology Blog, and it was having to do with a dengue antibody enhancement of Zika infection. I thought we were going to do the paper on TWiV anyway, so I thought we would wait and do it this week, because he had written, please read this letter tomorrow on TWiV. I said, no, we're going to wait.
Wait, maybe he said that in a different email, but the one he
sent to me, he sent me an email.
I thought he meant, okay, he said, please discuss this email tomorrow on TWiV. Okay.
Yeah.
But, but it starts out ADE paper, please discuss this email tomorrow on TWiV. So I thought he wanted us to discuss the paper.
No,
I see what you're saying.
He wanted me to read the email, but I thought we should just discuss it, put it out there, and then wait a week. Anyway, Vincent, I was recently made aware of your blog post on the paper discussing the possibility of dengue virus antibodies enhancing Zika virus infection. With utmost respect for you and your efforts on science communication, I was a bit disappointed by your blog commentary that was posted regarding this, quote, paper, unquote. First and most importantly, this paper did not go through a formal peer review process. Instead, it was simply published online. It was only screened for offensive and or non-scientific content. See below, and he posts the descriptor that you can find over at bioRxiv, which we know about, that it's not peer-reviewed yet. It's in the process of being. One would not immediately know this simply by looking at the paper. I find this process a bit disturbing. Second, the authors use the highly cell culture and neuroadapted strain of Zika MR766. While it is the prototype strain, scientists have to be careful to consider the Zika strain and temper their conclusions appropriately. Third, and probably the most offensive. There are several controls that are missing in this paper that make the findings difficult to interpret. Figure 1C and D lack a negative control. Where are their controls with mock-infected lysates? Figure 2. Without a positive control for Zika virus neutralization, the results are uninterpretable. How do we know that their assay was working correctly on that given day? In a later figure, the authors show Zika virus neutralization using dengue virus sera, but a nonspecific negative control is needed to appropriately interpret these findings. Figure 3, again, the authors did not include a nonspecific negative control sera. In the last figure, the authors measure ADE by PCR, which I feel is not appropriate, and you pointed out as well in your blog post. This also raises a serious ethical and scientific concern. Does this data exist in the scientific knowledge space if the work has not rigorously evaluated through a formal peer review process? Is this not one of the most integral aspects to rigorously evaluating one's findings? What happens if this paper goes through the peer review process and is rejected for publication? Would we be made aware of the reviewer's comments? I strongly disagree with research, especially of this importance and magnitude, being published through a server before it has ever gone through peer review. This can have damaging consequences if the findings from this research are incorrect and not representative of the current epidemic Zika virus strains. All right, allow me to respond, and then everyone else can chime in. Now, Mehul, I am a communicator, but I'm also a scientist. And I think you forgot. I am aware of how to review a paper and read it, and I understand it has limitations, but I found it convincing. And some of your controls are absolutely unnecessary. And this is part of the problem with peer review. As Ian Lipkin says, the main function of peer review is to delay publication of your competitor's work. Quite often, it does not accomplish anything. So I agree that some controls need to be done. We talked about that last week. But I thoroughly disagree that these are useless conclusions. I found them compelling, and I'm convinced at the results as they are. Now, the other aspect of this is preprint publication. Everyone knows this is the way that we're going. We've talked about this many times on TWiV. Harold Varmus and Ron Vail are all for it. Many people are doing it. The Zika publication spree is showing that it's working really well. I love the opportunity to have work early and not wait for 6 months for the journal to publish it. Although, of course, in the case of Zika, it seems to be shortened to a week now. So I don't think there are serious ethical and scientific concerns. In fact, as Ron Vail said recently, you post your first paper, and then you are allowed to post a revised version, which is the one that goes in for publication. So you can see how the data have changed. And I went to another publication server called PeerJ, right? And they posted a paper called Zika virus impairs growth in human neurospheres and brain organoids. And then that same paper was published in Science a few weeks later, virtually unchanged. Okay, so sometimes you don't have to make any changes. So I don't think that there's a problem with preprint servers as you do. I think this is the future. If you don't get behind this, you're going to be left behind. So I really disagree with most of your concerns here.
I would add that if every single one of Mehul's criticisms of bioRxiv apply completely to every scientific conference that I'm aware of.
Yeah.
Scientific conference abstracts are not peer-reviewed. They are submitted. Somebody goes and gives a talk, which is not peer-reviewed. Right. They present this stuff. And in many cases, the abstract, and in some cases, at very forward-thinking societies, they actually put up online multimedia versions of the thing, so anybody can go to that talk. Sometimes there's even a transcript or a write-up. So by this same logic, we would have to ban all scientific conferences because they're not peer-reviewed. But I think there's enormous value, actually, in having these preliminary results out there. And what people are now saying is, this hot, new, novel concept of open peer review is not a new concept at all. We've been doing this all along, but we've been doing it face-to-face orally at conferences. Now, the internet allows us to do this in a more structured fashion, and I think it's great.
I want to comment on several things. I think I fall between Vincent and Mehul in terms of the preprint server concept. I disagree with Vincent that everybody knows about it and that it is absolutely, going to be the way of the future. I think that's a bit strong. I have talked to several people and said, you know what bioRxiv is, and they don't. But if they're scientists and I tell them it's a preprint server, the word preprint immediately conjures up something in their mind as opposed to a peer-reviewed paper. And so I think there's room for people to put things up on preprint servers, and bioRxiv right at the top tells you that it's a preprint server and that things aren't peer-reviewed. I think maybe what stirred up Mehul, but definitely when I went back and looked at Vincent's blog post, what bothered me was the fact that the way the paper is referred to is just by highlighting the word describing some of the work in the paper with a hyperlink, so the text comes out in a different color. And if you click on that, then it takes you to the bioRxiv paper where at the top it says that, it's a preprint. But as we talked about last week, a lot of people don't click through on links. And a couple of people I asked, I just said, could you just go ahead and look at this blog post? And after you've looked at it, I have some questions for you. And 201, none of them had clicked through. They did not realize that the paper wasn't peer-reviewed and was a preprint. And so I think my point to Vincent was, and maybe Mehul was making this maybe not so directly, was that at least in several other places where I found the preprint referred to, they said, it was posted on the bioRxiv not peer-reviewed preprint server or something like that, that at least for the lay public and perhaps the popular press would indicate a little less stringency than a paper that's published in Science, Nature, or something like that, where we know it's peer-reviewed. Now, I agree that peer review isn't perfect, and maybe some of the controls that Mehul is asking for wouldn't be necessary, but we did talk about some last week that I think would have strengthened the paper and strengthened the conclusions. We also talked with Nina about, when you see something, you should ask, what's the source, what is the authority behind it, and also what is the N. And so some of my problems with the paper were that things didn't necessarily show me how many times they were repeated and what the N was. And getting back to the idea of the source, Vincent is well known now as a science communicator, and people read his blog and they take it as face value. And there's just no reference in the blog post saying this is a preprint. There are caveats here, that some people might want to see, more experiments or, something like that. So I agree, conferences, that's not peer reviewed, but people do get to ask questions, and people then do go to the bar or whatever and talk about some of those results in more detail. But people don't generally, at least to my knowledge, blog about them in a way that would be picked up by the popular press.
Right.
Well, it's going to start if it hasn't started already. Okay. I actually corresponded by email with the author while I was writing this to clarify some things, and including, did you do this experiment and that? And I was satisfied with it. And I put a link. I would argue that a link is fine if you want to see where it is. I'm not doing anything misleading by having a link to the original paper. Come on, that is transparency. I'm not going to say, why do I have to say it's bioRxiv? I never say it's Cell or Science. I just highlight it and link whenever I write an article. And I figured it's really the same thing. So why not?
I've talked to several people about that, and they disagree. They think that
well, because they're all in the old science model, right? They probably don't even read blogs, and they have no idea what the value is.
I would actually side with Kathy on that point, that it's worth pointing out in the text that this is a preprint, just as you would point out that it was a conference presentation if you were blogging about that.
All right. What does it mean to a non-scientist to say this is a preprint? Then I would have to explain.
That's why I think you have to add the caveat or the explanation. Just say a preprint, parentheses, meaning it hasn't been peer-reviewed.
Right.
That's all you have to do.
It's just an additional little reminder.
If all of you believe that is necessary, I will do it in the future. But I made a judgment call. And the tone of Mehul's article, I think, is totally inappropriate for that. And it overlooks the fact that preprint servers are getting more popular. It's not an ethical and scientific concern. The idea that the people are going to publish crap is total nonsense. for example, Ron Vail in his talk recently, one of the concerns about preprint servers, increased irreproducible or poor quality work. No, people don't want to put up irreproducible work on a preprint server. They want their best work there. They want it to be eventually published, but they're putting it up early so that people can see it. So I don't think these are valid concerns. I think
and also the preprint is not a cell paper in terms of your tenure committee or what have you. So people are not going to be gunning to put their stuff on a preprint server that's irreproducible or poorly controlled. It's more like a blog post. It's more like something that they want online quickly because they want the data out there.
Yeah. But they also
they did it first.
Yes, they get the publication date, and
yes.
Maybe that's what's bothering Mehul, that he's losing on the publication date or something for this. But Sharon's group has the publication date first of ADE with dengue antibodies to Zika.
Yeah.
I
certainly agree that Mehul is going way overboard with the critique here.
All right, so tell me, do you want me to put in the future, because I'm going to be blogging a lot about bioRxiv and PeerJ, you want me to put, this has been published on a non-peer-reviewed server or something like that? You think that's really important? I know you and Alan do. What about you, Dickson?
Well, I have an operational question to ask here. Let's say I'm an investigator and I've got a great result. I know it's going to be published sometime, but it takes a long time. So I'd like everybody to know about it now, and that's what this preprint server is all about, I presume.
In part, yeah.
You submit to both of these places, and you wait 3 months for your paper to get reviewed, and in 2 days, your same paper is up for everybody's observation. Is that correct?
Mm-hmm.
Right.
That's how this works.
Yes.
Okay, so everybody who reads those preprint news, it's news. before it's peer reviewed and accepted, it's news. Here's what we've done, and no one else knows about this except now you do, because I've just published it. But it's not a publication, right? So
it's an indexed publication, but it's not peer reviewed.
Well, the word preprint and reprint sound very much similar. Preprint and reprint. It sounds too close. It sounds like this is a review. This is a preview of a paper that's been submitted to. And if you put it in those terms, I would be all in favor of it. A preview of a paper that I submitted to. And then you know the whole line on it. So I submitted this paper to Nature, and I'm waiting to hear from them. But by the way, here's what I submitted. When I hear preprint or reprint, I'm thrown off by those terms.
Well, preprint to me, Dickson, means any manuscript before it's been submitted.
Understood.
In the old days, you would get somebody's hard copy and said, "Here's my preprint."
So the used car industry has a wonderful way of using the word pre. And you know it as well as I do. They call it pre-owned.
Yes.
That's called previously owned. That's the whole word, but they don't say that. They say pre-owned. For me, that means before owned. That is exactly what that means.
Preprint means before publication.
Yeah, I know that.
Yeah.
People are,
right.
but the common person out there will say pre-owned. That means that someone else has owned this before. Preprint, well, that means that it's been published before.
No, I think there's
it's confusing,
especially if you parenthetically explain the term at least a few times on the same blog. And this term is going to become more widely used in discourse about science, I think.
Yeah, well, then maybe we should discourage the car industry from using pre-owned.
We should also discourage the airline industry from using pre-board. But that's
indeed.
It used to be used cars, right?
Yes.
And they changed it to pre-owned. It sounded better.
It sounds like a pre-owned car. This is a brand new car.
Yeah, we've changed a lot of words.
Yeah, we have. So this falls into the gray area of.
So basically, my call was I looked at the data and I was convinced, and I thought it merited just writing about. If I had serious concern, and I noted my one concern, which was that virus yields in one experiment were not measured by infection but by PCR.
Yeah.
And I actually asked the author, and she said, we haven't done that, you're right. So I felt it was fine, and that's why I didn't do anything beyond.
So I see the value of this I see the value. I'm sorry.
You had additional information that you chose not to share in the blog, and that's fine. But for those people that only have the preprint and only have your blog, if they even know in your blog that you're referring to that and that you're convinced by additional data, that to me that's different. And I did see at least one popular press thing that pointed out that this was a well, no, it wasn't a popular press. Sorry. That just said, this is from bioRxiv, which is a preprint server for non-peer-reviewed, not formally peer-reviewed things. It doesn't mean that she didn't pass it around or you didn't read it or, and sort of peer review it informally, whatever. But I just am concerned that the lay public is going to take things like this, as, absolute truth, because they come from someone like you, that has a reputation and some gravitas here. And just adding a little bit more to it, I think, is truth in advertising and, isn't going to change the quality of your blog and just might inform certain non-scientist readers of your blog. Well, because as we know, there's plenty of people that read your blog that don't listen to TWiV and vice versa.
Yeah.
So the fact that we talked about the caveats on the paper last week isn't apparent to people who only read the blog post.
I mean, I don't want to go on and on about caveats in the blog post. That's not the point of it, right? The point is to make it as quickly as I can, right? And I think this is fine, and it's my judgment. That's why I did it. So, that's why I think people read it, because they say, okay, if this guy says this, then I'm likely to believe it. So I don't think I portrayed anything that was wrong, but I'm happy to put in the future this is on a non-peer-reviewed publication. Right. But the other thing I want to bring up here is that I don't think peer review is all that great anyway. We have, have done recently, Kathy, some of the Zika papers need controls left and right? So it's not the be-all and end-all to everything. And I really like this idea that we're getting this stuff out early. And I think this can evolve to a point where what goes up is really good because people know that they're going to get a lot of eyeballs early on.
Right. But I think peer review does provide an additional set of quality control, which is far from perfect.
Yeah,
but
it does. Yes.
It's an additional bar that you have to clear beyond just, your paper is not spam.
Understood. Yes. But I think going forward, we're going to see, at least in our field, of all these preprint papers that go up, we're going to see the next one, and we're going to see how much difference there are. Like the one I pointed out, the organoid paper, it was virtually unchanged from the Peer J version.
Right.
They didn't have them do anything. But as you say, Alan, at least it went through some kind of a check, right?
Right. And I also think that the amount that you'll be blogging off bioRxiv is going to fluctuate and will probably drop significantly when you're not writing regularly about something that's really, really hot.
Yeah, I think Zika has pushed it.
Out on bioRxiv and you'll say, well, wait till the paper comes out to even bother reading it.
Yeah, I agree.
Unless it's right in your field.
No, I think because if Zika is hot, that's why we're looking at bioRxiv.
And the volume of stuff that's on bioRxiv is growing and is going to eventually be, up at beyond +1 proportions. It's just going to be a tidal wave of stuff. You won't be able to keep up with it.
Do you think this will have an effect on the time of review of papers?
I don't think so. Why would it?
Well, because the results are already out there and everybody's reading them, and they're going to start talking among themselves in meetings and et cetera. And that paper still languishes on somebody's desk because they've got 15 other papers to review. And the next thing you know, it's 3 months down the road before you even get the review back. In this case, you're 2 days into it and you're online and you're out in front of everybody. So you don't think this will place pressure on reviews of other papers?
I don't think so. No.
Because
I think people.
I would hope it would.
Dickson, I just think people are busy and they get to reviews when they can. Nothing.
What I do hope it does is finally cut the stupid embargo system that we've got.
So Alan, that's a good question. If you have a paper on a preprint server, does the journal still put an embargo on it?
They might.
I guess so.
But it's already out there. I mean, the horse is out of the barn, and you're not gonna get the same sort of splash from it, which really I think the journals need to give up that habit anyway. Just put the papers out there and go ahead and promote them as you put them out, and if it gets picked up, it gets picked up. But
so operationally, how does this work once the paper is accepted? Does that
stays there? It stays there.
It stays there forever.
It stays forever, and you can submit the final version there as well. It gets a date. Yes.
Wow.
So Ron Vale went through all of this. And that's why I want to have him on.
Definitely going to kill the paid per view science world.
That's part of the point. Yes.
Alan, do you know in general? And it's kind of maybe too early, but do people like Carl Zimmer write about preprints, or do they wait for the final thing?
I haven't seen many people many science journalists jumping on preprints. A few reasons. One is that they don't generate press releases, so all the folks who are reliant on that stream for their news stories are not going to hear about these at all. The second thing is that the preprint servers are just not on the journal radar of most science journalists, and when they are, I'll go and I'll look at bioRxiv, and as I say, it's a rising number of papers, and it is already getting to the point where if I want to just read the table of contents of Cell, Science, Nature, PNAS, and a few other journals, and then go over to bioRxiv, what am I going to be looking through? I could spend days looking through just what's been published. So the amount of material there is very intimidating. And I think the final thing is, once people look at what bioRxiv is and they say, oh, well, this stuff isn't peer-reviewed, science journalists who still care about whether they're going to look like an idiot for having, raved about some research that turns out to be bogus, will look askance at that and say, well, I'll let this simmer a little bit.
Alan, that happens in the regular peer-reviewed articles.
Oh, of course it does. Of course it does. But then at least, critically, then there are other people you can blame.
Yeah, that's true.
For the physics things that are on the original archive, do science writers pick up from those, or do they wait until they're published? Do you know?
I don't really know the physics stuff. Physics proportionally gets very little coverage, which is not great.
Some of them are never published, but nevertheless they become part of the CV of the authors, and they're reviewed at tenure reviews. And that's the way physics has worked. But some of them do get published. Yeah.
Right.
In fact,
I was just trying, because I do remember Carl Zimmer saying that he was always a little disappointed, or is always a little disappointed when an author wants to talk to him after the work has already been published.
Yeah, right.
He did say that.
Right.
So that gives us a little bit of a clue.
Right. And that's, science journalists have already developed a certain skepticism about conference publications and presentations at conferences, and that's why you don't see a whole lot of people blogging about so-and-so said such and such at a conference. Because the understanding has evolved that, well, that's preliminary work. But
this is out there.
Somebody like Zimmer, his bread and butter is talking to somebody about something they're working on.
Yeah.
Getting that whole complex backstory put together, and really being able to tell the story so that when they do finally publish, he can be right on it with a really in-depth coverage. I
got the impression that Zimmer kind of liked this idea, right? And he wasn't limited by an embargo.
Right, and he and I certainly agree, and I think pretty much every science journalist agrees that we'd love to see embargoes go.
I mean, frankly, the journals, as we've talked about before, the luxury journals have such a stronghold on publication. I think it's absurd that we all feel that they hold the final word when, in fact, the decisions are often made not by scientists. That's why eLife is cool, because the decisions are made by scientists on the journal. But, you can have at Science and Cell, these are professional editors who are not in the field, and they're making decisions. I think that's totally inappropriate. And we are beholden to these people, and I just don't like that at all. That's why I love this preprint issued.
Vincent, I thought that the professional editors at these journals just were sort of a manager of a team, and the team actually does the playing on the field. So the reviewers actually have the final say as to
They don't
the
have
paper.
the final say. Absolutely not. I can tell you dozens of times I've made suggestions, and they publish it the next day because it's hot and they want to get it out, which is not the reason to publish something.
Or the author comes back to one of the reviewers and says, "You've got to publish this thing. Come on, what are you doing to me?"
Sometimes, I mean, it's all behind the scenes nonsense, and I just don't like the way that plays at all. I think the science needs to be standing for itself, whether it's hot or not. You review it if the experiments support the conclusions, you publish it. Right. This hot or not hot is absurd. And that's how these preprint servers can get around that, I think. I think they can take a lot of the wind out of the
other thing that preprint servers can do is that they can put some of the community feedback right on the paper. So you can have comments and I guess it's a preprint, but it's in a sense it's post-publication peer review because you put it out there. to get people's comments on it so that somebody, emails you and asks if you're going to do a plaque assay instead of PCR. And then that also is an advantage to the authors because they've got a preview of the kinds of things that are going to come up when they do submit it for peer review.
I just had an aberrant thought here. What if all scientific papers were submitted to Amazon and then they were open for criticism from the entire scientific community, and you were ranked, according to very good, good, not so good, horrible, and they added it all up, and then, you can read it, you can make up your own mind, everything gets published, but here are the reviews, and the reviews all came in from all over the world.
Dickson, in the end, the science that is worthy of being repeated will be reproduced and move forward.
Correct.
Really, so what does it matter? In the end, and there are many papers people will never pay attention to.
This is true.
But this, for example, the ADE paper we're talking about here, there will certainly be many other groups validating this and moving it forward.
No question.
So I don't think we need to have this cell science nature hierarchy at all. I wish it would go away. I think it's a pollution of the purity of science.
Listen to him. Listen to him.
All right.
Wait a minute. He's 2.5 feet above his seat right now, floating in space like a Buddha.
Look, we're lucky we have a platform here where we can express our views and people listen.
That's right.
And I appreciate that.
Yep.
And so I take advantage of it.
Yeah.
I let you know what I think, Dickson.
And just
so you do.
As an aside to that audience, we are eventually going to get to talking about viruses in this episode. Don't worry.
I think this is important, isn't it?
It is. Yes, it's a sort of a meta-virology thing.
Sure.
I just love these preprint servers. I'm going to submit a paper there for sure. Our next paper I'm going to send there. I think it's lovely, and I think it's here to stay. And frankly, if you're a scientist and you're not looking at these, you're going to miss out, because I really do think ASM is going to do preprint servers for its articles.
Look at that.
All right? And this is terrific. Anyway, Kathy, can you take the next
Speaking
email,
of
please?
Amazon.
Yes.
This is from Steve, who writes, "Hi, Vincent and team. Just thought you ought to see this. I checked out the Offit book on the Amazon.uk site and was quite sickened by what I found. The Offit book has just 2 reviews, both accusing him of making a fortune from, quote, big pharma, against which there are dozens of anti-vaxxer books, all with 4 or 5-star reviews. Anyone who looks for a book about vaccines on Amazon will, quote, only see that the, quote, vaccines bad view is the orthodox one. Rubbing it in even further, one of the 2 reviews criticizing the Offit book says we need someone to write a book with a balanced view. The flat earthers are way ahead of the scientists in getting their message to the public. One thing that could at least give the public a chance of seeing a balance would be to try and get people who really know the science to write proper reviews of both Offit's book and all the many recommended charlatan books. I don't really understand why these are allowed. They all ought to be contrary to advertising standards and be withdrawn as a danger to public health. Surely you could get the Twix community to start an Amazon balancing review writing campaign, at the very least by getting the Offit book that you all seem to admire, an overwhelming number of 5-star reviews on both sides of the Atlantic. A further point, which I hesitate to make, but which I must, is that there is a distinct sarcastic and superior tone in the majority of work that I've read and heard from what might be called the skeptic community, and that tone even comes across in the voices and comments of some of your own team on occasion. Some of the links that you provide for the balanced view are actually to some of the most sarcastic and superior writers and publicists, certainly those you refer to on this side of the Atlantic. I know that it is incredibly frustrating trying to reason with people who are too ignorant to know how ignorant they are, and who you know will never read any link to authentic information. But it is much better not to say or write anything at all if it can't be done in measured tones that don't sound superior and patronizing. I'm a, quote, skeptic, parentheses, I don't believe in anything, comma, but I honestly can't bear to read many of the people who think they are doing a great job of putting down bad science. Most of the time, they are just making things worse by alienating undecided people with their superior or typical doctor who thinks he could do stand-up comedy with lots of stories about quaint patients they've had tones. These seem to be a large percentage of UK doctors. Lobby groups like the Science Media Center and Sense About Science are, in effect, little more than a recruiting service for the conspiracy theory industry, which by all accounts is now very big and profitable. even though I don't believe anything from SMC or SAS, the two aforementioned groups, without careful checking, because they are not just promoting science sense. They are promoting science with regard to promoting British business, and do spin the facts, quote facts, accordingly. As David Tuller will tell you, they also promote some establishment popular science beliefs which are not yet supported by evidence. Some of their associates were familiar to environmentalists as media climate change contrarians before Spiked gave way to SMS. I'm pleased to say that I think Nina Martin came over as an encouragingly down-to-earth person to be going forward as a science communicator and teacher of science communication. If she can also pull off a sensible interview with Andrew Wakefield, despite getting targeted for abuse by the diehard conspiracy industry trolls, it will be a great thing to hear. But I do think there are an awful lot of science champions, in quotes, who need to be listening carefully to the likes of Nina and Kathy too, I think, because most of the science guys, in quotes, really aren't doing us any favors. Sorry to have to say this, but I've even seen factual inaccuracies on some of the outspoken skeptics' blogs, and when I've written comments with links to the correct case or data, they've just been deleted without acknowledgement. I detect far too much ego and testosterone for the good of effective science communication. Anyhow, I hope I can at least expect to see a few good positive reviews of Offutt's book on Amazon before long. And he gives the link to, I guess that's to Offutt's book. Keep it up despite the criticism. The last few podcasts have been especially good. Smiley face. Steve from England. And he says, the weather, typical changeable English spring.
I think of skeptics as the anti-vaxxers. So he's obviously...
They often brand themselves that way, yes.
Right. Now he's saying the skeptics are the real scientists, so I'm not,
No, I didn't read it that way.
But he says we're skeptics and we're arrogant, right? That's what he's saying.
No.
Except you and Nina.
No, I didn't.
He says the skeptics, like we cite, like I think that site that I like, Science-Based Medicine, he would say is a bunch of skeptics. I wouldn't label. I say the skeptics are the people.
That's how he's using the term. Is the skeptics are the people who are rationally thinking, and that's how the term is used culturally.
Yeah.
So I think that's the usage that he's using.
Okay.
I don't. So who are the science guys? Like, what's his name?
Bill Nye.
Bill Nye. Why does he say most of the science guys aren't doing us any favors? I don't understand.
Well, I do see his point that many of the responses to the anti-vaccine stuff come off as a bit patronizing.
I agree with that. I agree.
And it's very hard not to get that way when you're writing about that topic and you know that what you're responding to is complete and utter crap. There's a certain amount of frustration that's going to come through. But I think his point is sound, that we need to be wary of that and keep in mind how that comes across to people who are maybe undecided on this issue, not because they're stupid, but because they have been misinformed.
I always assume that they're not listening to TWiV anyway.
Well, possibly not. But
so we tend to be very sarcastic about it here. I agree. But that we're not talking to them. We're talking to an audience who is already on board with vaccines. Right. If I knew we had an audience of other people that were on the fence, it would be different. And that's why, Offutt and Seth Mnookin are good, because they have a balanced view and they don't get sarcastic. I understand that completely, but I'm not talking to them. But as you say, it's very frustrating because you have scientific evidence behind you, and yet everything you say is just twisted and thrown back in a different way. The thing
is that I get the tone of this criticism, and one aspect of it will serve to legitimize the opposite view of whether vaccines are good or bad, and that is if you treat them with respect and you have a good point of view, and I'd like to discuss this with you. If you go into the discussion with that attitude, you've immediately legitimized their points of view, and it's not a legitimate point of view. They have, so you're tempted to throw humor into it or sarcasm.
Nina said, look, last week Nina said at this movie.
Oh, she was great.
That people were yelling at,
Right.
It doesn't matter what you say, they'll yell at you.
No, Sure.
They just yell out liar, and then,
liar, yeah.
and that's all they need to do. And we see the same thing in the political campaign.
In a theater,
one person can call people liars, and nobody else can call people liars.
I think it should be against the law to yell liar in a crowded theater.
No, good. But I think it's also still important to distinguish between the very tiny, very vocal, hardcore believers who you're never going to be able to convince, and who are the shouting a liar, who are the ones paying to see this, the movies about this conspiracy theory they love, and the overwhelming majority of the general population who just want to know what they should do with their kid.
Yeah.
And unfortunately, that minority gets to write the reviews on Amazon, right?
Anybody can write a review on Amazon.
That's right.
They're the ones who do.
That's right.
That's why, to Steve, they're so imbalanced, right?
So I certainly hope that Steve is going to write a counterbalancing review on the Amazon UK site. I think I'm not sure that I can, because when I log in on my Amazon password, it's going to know that I'm in the U.S.
Well, I'll go and write something for sure.
The U.S. Amazon site, if you look up Offit's book, you find there are also some troll reviews like that. But they are outweighed by the rational reviews, and so that needs to happen on the UK side, ideally.
And what did we just see? That Wakefield is making another movie?
Yes.
Why not?
Well,
Got a career.
Somebody's going to make one about him.
Some Hollywood writer
yes.
said something that they might get on that and write a movie about him because they think he's an interesting subject, but which is really means nothing.
Yeah, there have been some biopics about people that maybe aren't thought as highly of as others. So, yeah.
Well, I agree that Nina is very calm, and so she's a good voice. She doesn't get sarcastic like we do. Maybe she's not as invested as we are or something. But I agree that she would be a good spokesperson, so I'm looking forward to her conversation with him.
Yes.
All right, we have one more follow-up here. This has to do with the same topic.
Right.
Dylan writes, Dear Triumvirate, I really enjoyed your latest episode featuring Nina Martin. It was interesting to hear from a scientist who has seen Vaxxed and can make more directed comments on its claims. During your discussion, the topic of the Tribeca Film Festival came up again, and Alan made a comment about Robert De Niro not being an anti-vaxxer. Well, I think this is true. I wanted to draw your attention to an interview he did on Today a few weeks ago, and he links to it. Although he does talk about a need to have safe vaccines, he also mentions that he thinks it was a mistake to remove Vaxxed from the film festival because it's, quote, it's a conversation that needs to happen, unquote. Unfortunately, some of his other comments seem more biased towards the anti-vaxxers. I was left with the sense, correct or not, that he does believe vaccines can be responsible for autism. In my mind, this interview undoes a lot of the good that was originally done when he pulled the film. After discovering TWiV a couple of years ago, I have now transitioned to listening to the entire TWiV family to get me through my daily commute. Thank you all for your tireless work to educate the public. Dylan is a member of the Tischler Lab at the University of Minnesota.
I have to agree with Dylan about the Today Show interview and Robert De Niro. I don't think he's convinced that vaccines and autism are not connected. He thinks there should be a conversation and that we need more information about this. Unfortunately, yeah.
Sounds like he needs more information about
that.
this.
We have to get Ian Lipkin to call him up, right, Dixon?
Right.
Well, that's what I don't know is when the Today Show interview was with respect time-wise to Ian's conversation with him.
Yeah.
Maybe Ian saw the Today Show interview and talked to him after that. I don't know.
All right, this episode of TWiV is brought to you by CuriosityStream, the world's first ad-free nonfiction streaming service founded by John Hendricks, who started Discovery Communications. And this is a library of 1,400 titles, 600 hours of content available all over the world, and you can play it on any platform that you might be interested in. And we think this is of interest to our listeners because it has a wide variety of science and technology content in addition to other things like history and nature and so forth. They've also just started launching 4K content, one of the largest 4K libraries on the internet. And they have things like Stephen Hawking's universe, Next World featuring Michio Kaku, and a film called Viruses, Destruction and Creation, which they talk about Zika virus, of course. There you go. They're right on point. Life on Earth, a series that explores our biodiversity. And Microbirth, a documentary that looks at research that links the way babies are born with health in later life. So you get real science shows, not reality TV science shows, which you can find everywhere else. There are monthly and annual plans, and they start at just $2.99 a month, less than a cup of coffee or the cost of one title on competing on-demand platforms. Check out curiositystream.com/microbe and use the promo code microbe during sign-up, and you will get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank Curiosity Stream for their support of TWiV. We have a few papers for you today that I thought were interesting.
At the 1 hour mark of the show.
Sorry. You want to just stop now?
No.
We will keep it.
These will be quick papers.
We can do quick papers. And I must say at the onset that none of them are from preprint servers.
These are all peer reviewed, and we all know what that means.
Okay. What does it mean, Alan? Oh no, we just discussed that. I'm sorry. I'm just joking. All right, the first is a paper in Cell Host and Microbe. Structures of the Zika virus envelope protein and its complex with a flavivirus broadly protective antibody. This is a paper out of China, Chinese Academy of Sciences, and also the Chinese CDC.
First author is Lian Pandai. Final author is George F. Gao.
And these individuals have been working on this broadly neutralizing antibody, which apparently they first described themselves a number of years ago, that neutralizes not only Zika, but also dengue viruses, all 4 serotypes, yellow fever virus, West Nile virus, and has been shown to confer animal protection against dengue virus and West Nile virus infection.
It's a murine antibody, which I had to go back and look after a while, so wanted to make that point for people up front.
So it's a broadly neutralizing antibody, and these typically target the fusion loop sequence on the E glycoprotein. E is the major glycoprotein in the envelope of flaviviruses, which is responsible for attachment to cells and so forth. So this and other broadly neutralizing antibodies tend to target that domain, and they tend to be less potent than type-specific neutralizing antibodies, which say would just do dengue 1 or dengue 2, and they target a different domain of this E glycoprotein. And what they do here is to produce the E glycoprotein without the transmembrane part in E. coli, and then they make crystals. So this is X-ray crystallography. Make crystals. They solve the structure of the E glycoprotein. And if you remember, I think a couple of weeks ago we talked about the cryo-EM structure of the entire Zika virus particle done by Rossman and Kuhn at Purdue.
Right.
And this is, of course, the E part of that, and it's pretty much confirming that structure as well. And then they mix the E protein that they've made with the monoclonal antibody and solve the structure of that.
And it's nice to get this protein, which is obviously a pretty critical protein for viral infection, in both the cryo-EM and the crystal structure, because now you've got 2 completely different methods giving you the same information. And and crystal structures tend to be higher resolution as well, although cryo-EM has gotten a lot better in recent years.
Yeah, crystal's tough because you need to get a crystal first. And
you need to get a crystal, although
tough.
With stuff that crystallizes well,
yeah.
There are established techniques and kits, and you line things up. And I was recently talking to a bunch of crystallographers and cryo-EM people for an article, and the cryo-EM folks said, "Oh yeah, we routinely try to crystallize stuff before we do it in cryo-EM, because if you can get a crystal, then you've got you've got your answer. You don't even have to fire up the microscope."
forgot to mention before we talk about this structure of the E with the antibody, they actually show that this antibody neutralizes Zika virus infection in cultured cells and also in mice.
Right.
They inoculate mice intraperitoneally, and when they give them 500 micrograms of antibody, they're completely protected. And if they give them PBS, they get a lot of death, 40% survival. So this protects infection of cells and infection in animals as well. So with appropriate humanization and clinical trials, this could be a passive vaccine for people. It could be like ZMapp, right? Perhaps, but it's going to be a while before that would happen, I would think. All right, so then they do the crystal structure, and you get a lovely picture of this monoclonal antibody, right? The two heavy and light chains forming the antibody, binding to the fusion loop of the E-glycoprotein. And there's a lovely cavity in the monoclonal that fits right on top of this fusion loop. So you get a very specific image. The interesting issue here, which is really the crux of the paper, is that in the native virus, as far as we know, and by the cryo-EM structure, this is very clear, the fusion loop is buried in the interface between the two E subunits, right? So this antibody wouldn't be able to just sneak in there and bind this sequence. So they speculate that maybe, a fraction of the particles have some. So the way this virus matures, the E-glycoprotein, as it's maturing in the cell, is sticking up. They call it a spiky appearance. And then as the particle undergoes acidification and maturation, there's some cleavages that occur, and then the E proteins sit down on the surface, and that hides the fusion loop. So they're saying maybe not all the E's go down, and some of them are sticking up on each particle, and that's what's grabbing.
Is that true for all the flaviviruses?
What true?
What you just said.
The maturation steps, yes. In fact, that's known for other flavies, not for Zika, because they haven't studied that for Zika. No. It's for dengue and yellow fever, West Nile.
So the crystal structure for them would look identical to this.
It's very similar, yeah.
Okay.
So that's the issue here. How does this antibody obviously is binding virus particles because it's neutralizing them, but how it gets at this fusion loop is a good question. And I think this idea that some of the E proteins are actually sticking up, maybe on each particle, there are a few stray hairs sticking up, and that's what it's exposing the fusion loop.
Right.
But that's basically the
Or you
paper.
could imagine it like a hinge, that as you indicated with your hands, and that maybe it's undergoing kind of conformational change
breathing or something.
all the time.
Yeah, sure. Breathing, yeah, that could be.
Breathing, thank you.
I think what would be nice now to have the complex of the antibody with the whole virus, right?
Right.
Yes.
I'll bet Rossman is working on it.
That would be a cryo-EM experiment to do.
Yeah,
yes.
and then you can see if the ones where they're bound are tilted up or something else is going on.
And how about camel antibody?
You love it.
What would they look like? I do. I can't help but think about that a lot.
You do? You think about camel
monomeric,
single chain antibodies?
Yeah. What would that look like?
Let's see. So the way this is binding, the pocket is a product of both the light and the heavy chains, right?
Yeah.
So I'm not sure a single chain would bind.
Well, you could test it in vitro.
Camelids, right, Dickson?
Yeah, exactly.
Yeah.
Yeah, you could make a single chain. But anyway, that's a very nice paper. It's peer reviewed. It's in Cell Host and Microbe, and the pictures are beautiful.
And it's open access.
Access.
Lovely.
So anybody can go look at the pretty pictures.
Great pictures.
And maybe this will end up in people. Who knows?
Yeah.
There's Zika in China now, so that's kind of a urgency for them too, I suppose.
Sure.
Next paper is also in Cell Host and Microbe, and this comes out of Brazil.
This is, part of a large, continuing, mysterious. How does that work?
This is a brief report. It's very short. It's a couple of pages, and it's from, what is this? Centro de Pesquisas René Rachu. I don't know where it is, though. It's in Fiocruz, but I don't know what institute. Anyway, it's the first author is... Kathy, can you pronounce it?
Dutra?
Dutra and Moreira is the last author.
Yeah.
And the title is Wolbachia blocks currently circulating Zika virus isolates in Brazilian Aedes aegypti mosquitoes.
Remarkable.
We have talked about Wolbachia before in terms of dengue control. I was going to look up the TWiV episode. It was a while ago, and it had to do it was work done by Scott O'Neill in Australia, who, by the way, was the first to find that Wolbachia, which are endosymbiotic bacteria. They live in the cells of other organisms, like I think 40% of the insects
It's amazing.
in the world have Wolbachia.
Parasitic worms.
That's right.
Parasitic wasps.
We did that on TWiV, actually.
Yeah, we've had some TWiV
Right.
episodes on Wolbachia.
In fact, there was a Zimmer article in the Times, which is quite nice, actually.
Yeah.
And he calls Wolbachia, the least appreciated creature on Earth.
It's a wasp egg.
But he talked with Scott O'Neill, getting the behind-the-scenes story. And I love this quote. Scott said, "It was just curiosity-based work. Total serendipity."
How about that?
The idea that having this bacterium blocks dengue infection. They just said, let's try it. And that's the way. That's great about science. We don't want to get rid of that. Please, let's have some exploration. Anyway, you put Wolbachia in. So Aedes aegypti, I believe, doesn't have Wolbachia normally, but you can make strains that have it in the lab, and then they get resistant to dengue infection. So this Wolbachia, you can release mosquitoes that have the Wolbachia, and because of its particular properties of cytoplasmic incompatibility and being transmitted by female mosquitoes, it can invade host populations.
So this is a gain of function experiment?
Yeah, it's a gain of function. You're adding Wolbachia, but you could argue that they're already in a lot of insects, right?
You could argue that.
But as we said earlier, Dickson, it's us that's mucking things up.
I tell you.
So they asked in this paper, they got a couple of Brazilian Zika virus isolates, and they they tested Wolbachia infected, and those were made in the lab. They have a line, or Wolbachia uninfected that they collected. And the way they infect these is that they take virus and mix it with human blood and feed it to the mosquitoes.
Yep.
Right, Dickson?
You bet.
What do you put it in a little...
It's a membrane. They can feed through the membrane, yeah.
Wow.
Yeah.
That's great.
Yeah, in fact, in labs where they raise mosquitoes, that's the standard thing. Is they'll have units of expired blood if they need to feed human blood, and they'll put it under this membrane just to feed the culture of mosquitoes that they're maintaining.
So, it should be said that they're only doing that to get the females to lay eggs.
Yes,
because they don't actually feed on them.
So Dickson, if you don't put blood in, they won't feed.
If you put sugar solution in, they will. That's what they actually feed on in nature.
Okay. So if you mix males and females and put them in the males don't pay any attention to the blood?
They don't have a mechanism for penetrating it.
What does that mean?
That is, the proboscis is not equipped to do that.
For penetrating the membrane, that is.
Oh,
yeah,
really?
Exactly.
Really? It's not, but they have one, right?
They have a proboscis, but it's not a...
And so even if you gave them sugar water, they wouldn't be able to get it, right?
That's
So what do males eat?
Well, they put these little slices of apple on the cage on it's got like a gauze cover that goes over the top of these rearing cages, and all of the males and females both stick through the gauze, and they feed off the juices of the plants. That's what they actually eat.
Got it.
So they do that with mouth parts.
Yes, they
Is that
do.
what you're saying?
They both do that through their sucking mouth parts, but the female has hypostyle that's capable of penetrating skin. She's got all the other heat sensing apparatus and all that kind of stuff that's good for them.
Yeah, Dickson. So when a female mosquito, alights on your skin,
yes,
and starts, what does it look for? A capillary?
It's got a heat-seeking missile, basically.
It probes.
It looks for the hottest area, which is near the skin, where a capillary bed is, and then it goes right for that.
And it can get into a capillary.
Yep, it can.
Well, and they also the proboscis, with a hypostyle, they penetrate and they'll probe around until they find a capillary that's suitable.
So typically, and this will be my last mosquito question, I'm sorry, how long would they sit on you and find a capillary? 5 seconds, 30 seconds?
It's
quick.
It's not short. The time period is not short.
And when they're doing that, do you feel them probing?
No, you do not.
You do not.
No.
Wow.
That's just and then once they do penetrate, they inject an anesthetic, so you'll never feel them doing that again.
Well, sometimes you see them sitting there, and then you get them before they can.
Exactly right. Exactly.
But if sometimes you hit them, and then you get blood on your skin, that means well, that could be your blood or someone else's blood, right?
That's right.
All right. Sorry. So here they feed the mosquitoes, and then they take the head, thorax, the abdomen, and saliva, and look for virus. So they have two isolates. One of them grows much better than the other, and they can. If one grows 3 logs more than the other, wow. 10 to the 6 PFU per mil versus 10 to the 3. By the way, they do plaque assays in this paper.
Yes.
Warms me heart. Just love it. I always ask people, why didn't you do a plaque assay? We don't need to do that. We do PCR. I feel like an old guy. But, sometimes you'd like to know the infectious titer. So at 7 days post-infection in the mosquitoes with Wolbachia, there's no virus. And this is
virus.
0 by PFUs.
Wow.
And in the other mosquitoes without Wolbachia, there's 0% of the mosquitoes, they sampled, I don't know how many, 20 per group, 0% had virus, and 65% had virus in the Wolbachia minus at 7 days. And then at 14 days, they get 10% with virus in the head and thorax, and 100% in the Wolbachia minus mosquitoes. So there's an effect of Wolbachia.
Big effect.
Interestingly, in the saliva at 7 days, you don't get anyone with virus. So I guess that's not long enough for it to make it to the
mosquitoes only live for 2 weeks. So
they only looked at, if you're looking at table 1, they say saliva infection was only examined for mosquitoes 14 days post-infection.
I see. That's
they
should have ND there, not a blank.
Right.
Oh, I guess 0 would be fine. Yeah. So at 14 days, the Wolbachia mosquitoes, 45% have virus in saliva, 100% without Wolbachia.
And how many viral particles do you think you need to initiate an infection?
I don't know.
Enough.
Idea. Do you know?
Enough?
Dickson?
No, I don't. I mean, in mice, they don't have a mouse model, but once they do have a
they have a mouse model.
Okay, so you can do this experiment in mice. You can.
Yeah. Why do you want to know how many particles you need?
Well, because I was thinking back to the West Nile story, because when the temperature is low and the viral titers in the mosquitoes are low, the chances are that people will not become infected even if they're bitten.
Mm.
So it requires a certain amount of virus in order to initiate an infection. And that's just why I was asking.
So this other strain, which grows poorly, they do the same experiment. 5% at day 7, 5% have virus in head and thorax with Wolbachia. 95% without Wolbachia.
So this is an experiment that requires like a P4 facility in this country.
This one?
No. If you were to do it here, you'd be worried about escapees.
No, you don't need P4.
No.
I'm going to tell you now, if you work in any building that harbors a mosquito colony, you'll find mosquitoes in your office.
Yeah, we have a colony here in microbiology, right?
A mosquito colony?
Yeah, Fiddock grows malaria in
Does he
mosquitoes.
really?
Yeah, but not the human malaria, the other one. What would it be?
The mouse malaria.
Yeah, what is that?
That's a culisine mosquito. See, there's no danger at all for humans because that doesn't transmit human malaria.
Yeah, okay.
The anophelines do.
But I think you don't need to be a self-board.
No, but I'm saying if you're going to work on Zika in Aedes aegypti, I bet you if you got that through the IRB, they would require at least a P3.
Anyway, there's a substantial effect of Wolbachia, right? On
huge.
Mosquito. And, it's not 0, but you may not need 0 to reduce
right, that's what I'm saying.
Right. That's why you asked.
That was actually why I asked.
Sorry, Dickson.
And I'm just a fly on the wall over here.
You're not a fly. Don't you have the notes in front of you?
Of course I do.
You're a mosquito on the wall.
Yeah, that's right. That's a fly, by the way. Mosquitoes are flies, by the way.
Yes.
Mm-hmm.
Just so you'll know what an angiosperm. Whereas angiosperms are flowering plants and mosquitoes are dipterids.
I just wanted to be specific.
I know.
Right? They also looked at RNA in these mosquitoes, and I don't know, they have dot plots where a dot is each mosquito, and
right.
These, so, I mean, with one virus, with Wolbachia, you get very little, you get no virus in the head, but lots of virus in the head when there's no Wolbachia. But then if you look in the abdomen, you get a whole bunch of mosquitoes with no virus, with no RNA detectable with Wolbachia. But then there's a whole cluster in the middle with some RNA, so it's not all or none.
So the question arises,
yes, sir,
when the mosquito becomes infected.
Even in the saliva. Sorry, Dickson, let me just finish.
Yeah,
Even in the saliva, without Wolbachia, you get a range in terms of copies of RNA from 5 logs to, say, 8 logs. And then with Wolbachia, you get a whole bunch with no detectable RNA. Then you have a whole bunch with 5 logs of RNA. So it's really strange how you get 2 populations there.
So
but again,
the hemosyl is filled with virus particles? That's what you're saying?
This is saliva.
Yeah, well, that's contiguous.
Well, yeah, that's what they do. They eat they take a blood meal, right, Dickson? Then the virus goes into their gut, right?
Yeah,
and then it gets into the hemocoel.
Well, it infects the gut cells first.
Right. Gets into the hemocoel, then it goes to the salivary glands, right?
Because, I don't know, does it?
Yeah, that's the idea.
So the viral titer that they ingest, some of that goes to the salivary glands.
Well, I don't think it's the input. I think it's replicating.
I do too. So that's why I'm asking about the presence of free viral particles in these animals.
Yeah, that's the first experiment. They did that. They look in saliva and in body parts for virus particles. So with time, it goes up, which is indicating that it's replicating, right? But that's the general scheme, right, Dickson? They take a blood meal, it goes to the gut, replicates in the gut cells, it gets into the hemocoel, and then travels to the salivary glands.
Or the salivary gland cells get infected.
Is that possible when
Absolutely.
you take a blood meal? Yeah.
Every single cell is possible.
Wait, when you take the blood meal, it can go right to the salivary glands?
Well, no, it goes into the hemocoel, which bathes all of the organs.
But wait, Dickson, when they take a blood meal, doesn't it first go to the gut?
Yes.
Okay. So then it has to go through the gut
not
to the hemocoel.
So let's say it goes into the gut, and then it infects the gut cells first.
Got it.
Then some of the gut cells rupture and release virus to the hemocoel side.
The hemocoel is the circulatory system.
Right, but it's an open circulatory system.
Right.
So every organ is susceptible to infection.
Got it.
Including the salivary glands.
All right, so it's like open access.
Exactly.
Yes.
Dickson, is there any case where a mosquito takes a blood meal and then goes bite someone else and then regurgitates and infects them?
They don't regurgitate. Not like
Mosquitoes
fleas.
or like mice, they don't throw up.
Or fleas that have plague and then they have to contain. No, I don't think so.
No, in fact, most mosquitoes, the first thing they do right after taking a blood meal is immediately land somewhere.
Exactly.
Because they can barely fly as it is. And then when they're full, the blood meal doubles their mass or something, and they immediately just go and they land somewhere and they sit there digesting the blood meal. And there you can find cool videos of this. They'll secrete the water from the blood over time.
They plasmapherese it.
Plasmapherese the blood.
That's true.
The water will come out. They'll pee that out, and then they'll take off and digest the rest of the blood. But they won't feed again until they're.
How long does that take?
At least 2 days.
Really?
Yeah.
So they go somewhere safe, right?
They try to.
They try to. They go somewhere. They go somewhere dark.
In the tropics, in less developed countries, residual DDT spraying just on the windowsills of the open windows is enough to control malaria.
Right.
Because that's where they would go.
That's where they land first.
Or tequila.
Not for the mosquitoes, though.
It's interesting. They rest.
They do. They have to. They're almost flightless birds at that point.
And so while the women are getting the blood, what are the guys doing?
They're out there having fun among the flowers.
And I can call them guys because they are guys.
You can. They're having a hootenanny. They're
Dickson, what do they do actually?
What, the males?
They do one thing.
They do it well too.
Really?
They find females.
They inseminate. That's what they do for a living.
Okay. All right. So are we going to release Wolbachia 80s
Why not?
for control in Brazil?
Why shouldn't
I don't have a problem with it. Does anyone have a problem?
No.
That's.
I think it's okay.
I mean, what could go wrong?
Save that phrase.
Watch this space.
Use it again.
Which, by the way, we could raise with the previous paper too, because you think about you're putting a broadly neutralizing flavivirus antibody, and we've already talked about what that can do to dengue and Zika.
Well, yeah. In fact, the paper we discussed last week, they were monoclonal anti-dengue antibodies, and they could enhance. Yeah. So it's not the. Yeah, but I think this would be a nice idea to release these and get those.
Oh yeah.
I think that an interesting question. There are many. How much do you have to bring how much of the population needs to have Wolbachia to be effective and so forth? Right. I think it's fascinating stuff.
And would it be effective? Would this actually be enough?
I think it's really neat.
Persist.
That's right.
Okay. I thought just 2 quick things. One, there's an article in Science by John Cohen. Zika vaccine efficacy trials could start in 2017. This is Tony Fauci. He says NIAID has been working on a Zika vaccine starting in September. Wow, they started early. And they plan to begin tests in 80 people. Sorry, I don't know when.
No, it plans to begin
in September. That's it, right.
They didn't start working on it in September.
Wait, NIAID, listen to this sentence. I'm not sure it's a great sentence. NIAID plans to begin tests of a vaccine made in its labs in September in 80 people. So
yes.
In September, it's a little vague, right?
Right. Did they make the vaccine in September, or are they beginning the test? I think they're beginning the test.
So I'm really curious. He doesn't say what kind of vaccine it is. This is Tony Fauci.
It's got to be based on the yellow fever vaccine, don't you think?
It could be.
Presumably something NIAID researchers have been working on, but we don't know.
Yeah, so we don't know, because to make a brand new attenuated Zika vaccine would take longer. Starting with just Zika virus. So I'm curious.
But you have an epitope to aim at, though, right? The common one aimed at?
The Z protein, it's got a number of neutralizing epitopes, yeah. But this, if it's based on the yellow fever backbone, it would be a replication competent vaccine, right?
Of course.
Anyway, so I'd like to know what that is. So hopefully we'll find out. Maybe at ASV, right, Kathy?
Maybe.
At the special Zika sessions.
Maybe.
One more snippet. This is pretty funny. Well, I don't know if it's funny, but it's an article in The Conversation. It's called Carpageddon. What you need to know about the release of carp herpes in Australia. So apparently, this is from La Trobe University. Apparently, many years ago, they brought carp to Australia for farming purposes, and then there were floods, and they got out. Right. And invaded the rivers, and now they're all over the place.
Because it's Australia.
How strange.
Yeah.
Who would have ever thought?
So
what could go wrong?
What is it with Australia? They bring things in, and then they overrun the rabbits, right?
Well, they brought themselves in first.
For hunting purposes.
Yeah. Well, it's because it's an ecosystem that evolved on its own, and so it's highly susceptible to this.
So there are too many carp, apparently. These carps can each lay 1 million eggs a year, and so they're stuffing the waters and polluting them, and
Some rivers, they estimate that 80% of the biomass is now carp. Which is insane.
True.
So they want to use a carp herpes virus and then release it, and it will kill them, many of them. Yep. And this has been approved. And $15 million is going to be put into this. And what
do you think will happen, Vincent?
You get a lot of dead carp.
And?
Then the resistant ones will begin reproducing.
That's exactly what I wanted to hear.
But, one of the interesting problems is you're going to have a lot of dead carp, and they have to get them out before they start rotting. Otherwise, that's even a worse problem. So they have a plan to do this. Dickson, you need some cheap carp? You could probably buy it.
It's a mess. No.
Actually, they said they can't send it elsewhere. It would cost too much to freeze it and send it somewhere else. So they have to.
It's a popular but very low value fish.
It's true.
They're going to use it as fertilizer, right? Pet food.
Yeah.
Well, so they don't really have the plan yet.
They don't?
Large part of the $15 million over the next 2 years,
to figure out
she thinks will be to figure out what they're going to do with this one big time carp kill.
Do we know when they're going to release the virus? By the end of 2018. Okay, so that's a ways off.
I have an alternate plan. They should hire a 5-star Michelin chef to come in and to create dishes based on carp.
All you have to
absolutely
do is
unbelievable.
Google carp recipes.
Yes.
There are tons and tons of recipes. I did this right before the show. And, deep frying is apparently a popular choice. But then there are a bunch of other strategies. You fillet it, you score the flesh so that the heat and the cooking oils will get in and soften up the little bones, and then you cook it. And there's also a joke recipe that I heard years ago that you take the carp and you attach it to a board, and you marinate it and everything, and you bake it for about 3 hours, and then you take it out and you throw away the fish and eat the board.
Exactly.
Exactly. Yeah, the Australians resist eating carp because they think they're junk fish, they taste muddy, and they're too bony.
So why
above.
Why did they bring them in the first place? They didn't.
Somebody else did.
Who?
Maybe the Asian community.
Oh, I see.
Yeah, they are there. And if you look up carp recipes, you will find they're certainly Chinese food recipes where you can get
tons of it. And in New York City, they used to import carp to New York, and it used to be a portion of gefilte fish making. And in order to make the carp taste good...
Because they're gefilte feeders.
That's exactly right, Alan. Thank you so much. That joke has been used so many times.
I know.
So they used to keep these carp alive in their bathtubs. And overnight, the muddy flavor would dissipate and go out of the fish. And another way of getting rid of the muddy flavor right away is to marinate them overnight in a dish of milk, So you make a fillet of the fish, and then you sort of let it sit in milk overnight, and it completely changes the taste of the fish. So there are lots of ways of handling this problem.
It is fixable, yes.
It's fixable. So, here's a potential barramundi-like fish. I know that people in Australia would cringe if I ever said that on the air. I just did, because that's their most delectable freshwater fish, I think, is the barramundi. So the carp could easily take their place if they learned how to cook them properly.
Dickson, what do you think of this idea to release a herpes virus to kill them all?
Well, I'm a little bit worried. Kill them all, right? I'm worried that it'll affect other local indigenous populations of fish, for one.
Yeah, they claim that won't.
They tried this with rabbits.
Yes,
that didn't quite work out the way they wanted it.
But they did,
reduce it.
As this news article points out, they did significantly reduce the rabbit population, and it has stayed much lower than it was at historical levels.
Yeah. It's true.
You're never, ever going to get rid of all of the carp. You're not going to be able to eradicate a species like this once it's been introduced. But I think if you can reduce it by a huge percentage, and then you get a stable population at a much lower level, it would just become part of the ecosystem.
Right.
If you click through to the previous blog post, she does describe that this herpes virus has no impact on other native fish, yabbies, and trout. It can't affect mammals, amphibians, or reptiles. Right. But the bad news is that it will affect ornamental carp, which are koi.
That's right.
So that's a problem. But
they are valuable.
Then she says, the solution to that might be just like vaccinating your pet rabbits against calicivirus. Maybe you would then somehow vaccinate your koi against the herpes
and
virus.
It shouldn't be too hard to keep the pet carp separate from the wild ones, because they are koi.
Yeah.
Alan, it's a slow news day out there, isn't it?
She says, and no, we didn't kill all the rabbits, just like we will not kill all the carp.
Right.
But we should kill all the lawyers, right?
That's what Shakespeare would like us to do.
Didn't Shakespeare say that?
It did.
Now, why did
that line is actually spoken by somebody who's trying to get a mob to
really
anarchy? Yes.
So they had lawyers back then,
right?
Oh yeah.
But it was obviously that line was inserted for to please the crowd,
and those lawyers were always carping. So I mean,
yes.
Arg. I can't outdo Alan. I'm sorry, I didn't even mean to try. You can delete that, Vincent, when you.
No, I don't delete.
No,
I don't delete. Only. Telling Alan to lower
the other day, Fred Alt gave a talk here, and the computer went down for about 2 seconds. And
he didn't know what to do.
I know, but I was so tempted to say, you shouldn't have hit Alt Delete.
Dickson, you should have.
I didn't.
That's very good.
I missed my big moment. Maybe he'll listen to this podcast.
This week, I want to let you know about the annual meeting of the American Society for Virology. This year's ASV meeting is being held in beautiful Blacksburg, Virginia, on the campus of Virginia Tech University from June 18th through the 22nd. The local organizers, Drs. X.J. Meng and Zach Edelman, and their committee are arranging this great venue for the meeting. The keynote address and plenary symposia were arranged by ASV President Grant McFadden. This year, the keynote address will be given by Dr. Stephen Russell on oncolytic virotherapy, followed by 5 days of symposia, workshops, poster sessions, and state-of-the-art lectures. You can come listen to Dr. Joe Handelsman talk about leading the White House Office of Science and Technology Policy, be part of a live TWiV audience, and enhance your presentation skills at the Career Development Workshop. Also this year, ASV is hosting a special Zika virus oral workshops and poster session. ASV has issued a call for late-breaking Zika abstracts with a deadline of Monday, May 16th, about a week from now, so get them in. I'm sure you're all working on Zika. Early registration for the meeting at the reduced rate ends May 20th. Go to asv.org or asv2016.cpe.vt.edu for more information. I hope to see you there. We'll all be there doing a live TWiV. All right. Shall we do picks?
Sure.
Can do more email next time. Dickson, do you have... No, sorry.
No.
I'm saying it wrong. Dickson, what is your pick?
Well, I do have a pick, actually. It's a video, computer-generated set of cloud conditions over the Earth. And you can click on this thing, and you can get cloud maps And you
cool.
It's a very cool site. It's hard to describe it unless you're a meteorologist. So our show features a lot of meteorology. So I thought one on clouds would be a good thing to have. And I saw this and I was fascinated with it. You can play with it for hours.
How did you find this, Dickson?
I just, I won't tell you, but if Yahoo News ever goes offline, I'll be stumped.
It will.
I know it's
not going to stay forever.
I know it won't.
You're in big trouble. And
that's how I found it.
And it's kind of old school Yahoo, you know?
No, I like.
I wish you would update your news gathering.
Oh, I read the Times also. Come on. I watch PBS.
That's not what I mean. That's not what I mean by upgrade. Okay, that's pretty cool.
Yeah, it is nice
lovely,
and very pretty, beautiful colors, and a lot of work went into putting this thing together.
I like the website. It's very nice.
Yeah, it's
clean.
It's clean. That's exactly right.
I wish I had a website like that. I tried, but lots of issues. I'm not a coder, you know.
Your website looks okay.
It's fine.
Okay. It's okay.
It's fine.
There's always some issue that it crops up.
Well, it's WordPress, right?
Yeah, it must be WordPress, right?
There's always everybody ends up there, and there's always stuff to deal with. But
what's better?
Well, I switched to using a static blogging engine, which requires work in a different way. I'm not entirely sure I'm pleased with that. I may switch back, but
there's an open source blogging engine which gets a lot of good reviews. I think
if you use
Ghost.
If you're self-hosting the WordPress blog, that's the scripts behind that are open source.
Yeah, that's true. But there's another completely open source. I think it's called Ghost, which is supposed to be great, but I've already put a lot of work in, so I don't want to do it again. Anyway, thank you, Dickson. Alan, what do you have?
I have, what did I check? Oh, yes. This is a paper from MBio by a couple of folks we've talked about and I think talked to in the past, Farrokh Fang and Arturo Casadevall. They have a modest proposal for research funding. And this is not a joke.
Eating all the children? No, that's not going to work.
No, their idea, which they first put forth in, I guess Arturo put this forth in a letter to Science right around the beginning of April, and I thought, okay. And then they've really fleshed it out here. It's MBio, so everybody can go and read it. It's a short explanation. Their point is, we've got all the funding gets tighter and tighter, and pay lines are getting smaller and smaller. So a smaller and smaller percentage of the grants are getting funded. And we now have ample data to show that study sections are actually really good at selecting about the top third of papers or of grants. But they're really bad at distinguishing who's going to do the good science within that top third when you get to those finer and finer divisions. So if you're only going to fund 10% of the grants, what you're left with is a process of bad decision making that's going to take the 30% that should be funded and whittle them down to 10% who are, names they recognize and entrench other biases or research they recognize. So the proposal here is continue with the study sections, but have them only basically do triage. Pick the top third of the proposals. From that set, figure out what the budget is and say, okay, we can fund this many grants, and choose them from the approved grants by lottery.
Yeah.
And their random table of numbers you're going to pick.
Their point is, right. It is no less accurate than the system that we have now. It's fairer because everybody who gets into that top 30%, meaning you've got a scientifically valid proposal that could work.
1 in 3 chance, right?
It's not a 1 in 3 chance. If you write a good grant, it's virtually guaranteed.
Oh, okay.
But if you write a crummy grant, you're not going to get it in. So you've got the first screen. You've got, the study section is making sure that everything is valid and could work and is an appropriate thing to fund. And once you pass that, you're entered in the lottery. And if you win the grant lottery, great, you got funded. And if you don't win the grant lottery, you don't have to beat yourself up about, maybe I could have written my grant better. But the lottery would be fairer because it would not rely on seniority or race or gender or all the other subtle biases that come into play when you start whittling down the pool that far. I think it's a great idea.
It's interesting, and the connection to lottery and another word that I've heard used a lot when people talk about putting their grants into study section, that is that it's a crapshoot.
Crapshoot. Yes, that's right.
It just, if it already is, then why not make it one that's
Crapshoot.
fair?
And their point is, right now what we have is a bad lottery. Let's make it a good lottery. Let's make it an actual lottery system that is fair, because right now we have an unfair lottery system.
That would ruffle a lot of feathers.
It would ruffle a lot of feathers, but it would also provide a really good rhetorical tool in going and talking about science funding, which is that the NIH and NSF can go to Congress and say, here's how many good grant proposals we got. Here's how many we were able to fund.
Right.
Yeah.
Rather than saying, okay, well, thank you for that tiny amount of money. We'll fund what we can, and we'll try and pick the best ones. But to be able to go and say, look, we've got all these great proposals here that we can't fund.
Yeah.
The other thing that it would do is for philanthropic organizations that want to do something about this, they would have a pool of pre-screened grants that they could choose from. So...
That's a very good idea.
Yeah.
So if you go to the MBio page, where there's, if you click through to the MBio page, over on the right is the image for the current issue. And the image for the current issue is a bacteria phage of the Podoviridae family, constructed from Lego.
Yeah, it's cool.
Oh!
Nice.
So,
The Bushmans, yeah.
yeah.
Okay.
Very nice.
Okay, I'll get to work.
Yeah.
I'll get to work.
Yeah, you should get to work.
Okay.
So, Kathy, when study sections review grants, what's the cutoff for triage percentile? Do you know?
I think it's roughly 50%.
Okay.
So half of them get written reviews, right?
Right.
So what about taking the ones that didn't get funded and funding them next year?
But then you have more.
And what do you do?
Well, then you have a 2-year cycle rather than a
And
1-year cycle.
And
And then
it's
you have a 9-year
scale
cycle.
science, because maybe they've already done it.
No, I hear you.
Yeah, I don't know.
Right. I mean,
So
if they were
this
good
is-
this year, why wouldn't they be good next year?
This is a good idea, but I can't see Congress allowing this to happen.
I
right?
don't think they would.
Well, it's not going to happen, but I think it's great as a concept. to have.
I do. I agree with it. I think I would not mind having part of a lottery, right?
Yeah.
But I
Sure.
just think it's never going to happen. It's too bad because Congress will never allow a lottery of any kind except the draft, right?
Right.
They do a draft lottery, right?
Well, actually, I don't know to what extent NIH would need congressional approval for this, because there would still be a peer review process for the grants.
Yeah, but they may not need approval. But when they do it, and Congress says, "What the hell you're doing," that may
Well,
be.
they would answer to it, but then they can explain.
Yeah.
We don't have enough money to distribute, so we're distributing the money we have in the fairest way possible to the good grants. And we can only say which half of the grants or which third of the grants are really good.
Yeah,
So
okay.
we're not going to.
I would like to hear someone from NIH respond. Remember the lady Rocky something?
Mm-hmm.
She's gone, but someone of her level.
Well,
right?
there's a new person, and his blog is called Open Mike. His name is Mike something.
Okay, he should respond to this, right?
Mm-hmm.
Okay, Mike, respond. I know you're not listening, but
You say that to me all the time.
what? That you're not
I
listening?
know you're not listening. Of course I'm listening. I'm not understanding, but I'm listening.
Kathy, what do you have?
I chose something that was on the cover page of the Science Times and the New York Times. And it's an article about Eugenia Cheng. And she combines her love of math with a love of baking. And she's a theoretical mathematician who works in a rarefied field called category theory, which is so abstract that even some pure mathematicians think it goes too far, she said. But she's winning fame as a math popularizer. And so the top half the picture shows this Bach pie that has something relating to knot theory and chocolate. So the CH of Bach is the chocolate, and I forget what
banana.
the BA is. Oh, banana. That's right.
Yes.
Yeah. And so she even has a book, which I have to admit I haven't read, but I have read the whole New York Times article, so that's what I picked. And her book is How to Bake Pie, and it's, of course, the Greek symbol pi, an edible exploration of the mathematics of mathematics. And so it's just a really fun article to read. And at the bottom of it, then they have the recipe for the Bach pie. And I just thought it was fun for people to take a look at.
That's great.
I come at this honestly because I have at least 3 cookbooks that are about the science and chemistry of cooking, and this just feeds into that interest.
One of them is very expensive, if I'm not mistaken. I almost bought that for my wife. It had the chemistry of cooking. It was like $600 or something.
Oh, that's not one I have. I have 3 others that were much more affordable.
I backed off of that one. You know, Kathy, are you familiar with Alton Brown's television show on cooking?
No.
Oh, I'm going to pick that next time. It's called Good Eats. I'm not kidding. He uses scientific equipment all the way through his cooking. Every bit of it is weighing and tearing and, making sure that how to pipe pastries and stuff. It's all done by the scientific method, and he's quite entertaining, actually. Remember, I once told you that he was picked as the plenary lecturer for the ASC meeting.
Cool.
The ACS, I'm sorry, ACS.
Okay, got it. Okay. Yeah.
Anyway, that's a great pick.
Yeah, I like that.
Yeah, there's an interesting thing in here. There's another math popularizer, and his name is John Baez. And yes, he's a cousin of Joan.
Really?
So he said, Eugenia has gone all the way in. She's trying to explain math to everybody, with or without pre-existing expertise. And I think she's done wonderfully.
So can we use cooking to explain viruses? I guess you could just cook virus cookies, right? Which people
Right. We've seen virus cookies in the past.
Yeah.
And cakes. Yeah.
Yep. My pick is very, very self-serving. Right? I apologize. You don't have to follow it or read it. It's a new blog I've started over at ASM called Zika Diaries, where I just write about every week our foray into research on Zika virus. And it's sort of a behind-the-scenes thing, how you get things done. And I will be writing about getting scooped and being sad about things and all that. It's a personal diary on, we just started to work on this virus like many other people, but I thought it would be fun to write about it, and there's a cool picture every week that we are taking. And so Zika Diaries.
This is great.
I thought it was. They asked me if I would do this, and
so
I said sure. I got a lot of free time, so I figured do that. But it's fun. And also, so where Zika Diaries is, in fact, they have a couple of other ASM blogs, and one of them is called Blog Phase. You get it, Dickson? Blog Phase by the CEO, the new CEO, and I picked one of his pieces a while ago, which was about preprint publications and how the ASM is into it, and he's into it, and so he writes a blog every week also, which is good. Blog Phase. We have some listener picks now. 1, 2, 3, 4 people. They all picked the same thing. This was a very popular pick. These are virus trading cards. They're not really cards. They're online because they're animated. And I think this person made a couple of them, and they have viruses spinning on the cover. One of the students in my class actually showed this to me once, and then Steven saw this and thought it would be a good listener pick. He gives a boing boing link. William thought you would enjoy these. He gives another link. Norma and Maurice, hello Twivsters. Thanks for your wonderful podcast. I look forward to every episode. I saw these cool virtual virus trading cards and thought others might like them too. And then Tom writes, in case you haven't seen these virus trading cards, they're a bit of a techie tour de force, but there is something beautiful and something unsettling about them.
Apparently, if you have a 3D printer, you can print these.
Is that right?
Wow.
Yes.
Oh. Wow.
Okay.
So the first person, Stephen, he wrote also, I have a total of about 5 hours of commute time a week. If each TWiV episode could be 5 hours, that would be great. I really don't understand people complaining about TWiV being too long.
We're working on it.
No, I think 5 hours is too long.
That's right. We could read Moby Dick on the air.
No, we want to do original stuff. All right, that's TWiV 388. You can find it at iTunes, microbe.tv/TWiV, and also at Google Play Music. And you can send your questions and comments to twiv@microbe.tv. We do have a backload of emails, so if we haven't read yours, we will get to it. Whether it be big or small or long or short, we shall read it at some point. I just don't know
That's
when.
a backload of email, not a front loader of it.
That's right. And since we're coming up this summer now with some travel related episodes where we don't do email, so the front load will get even bigger. Yeah. Dickson Despommier is at verticalfarm.com. Thank you, Dickson.
It was a pleasure.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
And Alan Dove is at turbidplaque.com. He's also Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.